Okay, so you should have this diagram of a nephron, and we're going to add some labels to it. Now, the nephron itself is this part here, so I've drawn part of it here. I haven't got enough space to have this big loop bit, but you can imagine it's going back down the page there. So each kidney has over 1 million nephrons, and the nephron is responsible for filtering the blood and for the osmoregulation. So there's a number of different parts. The first bit of the nephron is this cup-shaped structure, and it's really like a bowl or a cup, and that's called the Bowman's capsule. Okay, and then we've got this coiled, curved bit of tubule, which is called the proximal convoluted tubule. Now, it's a tube, hence the name tubule. Convoluted, if something's convoluted, instead of going from A to B, you go on a sort of windy, bendy route. So it's convoluted in that it's all twisty and windy. Proximal, it's in close proximity to the Bowman's capsule. So that's the proximal convoluted tubule. This big loop structure is called the loop of Henle, named after Mr. Gustav Jacob Henle, I think his name was. He was actually a German guy. It's a big loop that goes down and back up again. Then we've got another convoluted tubule, and this is called the distal convoluted tubule. I haven't got room to write it all out there. Distal convoluted tubule, really from there to there. And then the fluid will flow into this structure here, which is called the collecting duct. Now, I don't have space to write that. The collecting duct, you'll see there'll be tubules or nephrons coming from into there and there. In other words, it will collect the fluid or the filtrate from a number of nephrons. Now, in terms of where this would be, if we look at the structure of a kidney, the nephron would be starting, Bowman's capsule is in the cortex. So if I draw this cup-shaped structure and then the proximal convoluted tubule, loop of Henle, distal convoluted tubule, would look something like that. So most of the nephron is in the cortex, the outer region. The loop of Henle is pretty long and it stretches down into the medulla, the middle region. The collecting duct, as we said, you would have a number of nephrons flowing into the collecting duct. Collecting duct will take the fluid into the pelvis region. You'll have many collecting ducts flowing in there. So basically it's urine now, and it leaves the kidney in the ureter and then it goes to the bladder. So if we look at the structure of the nephron, it is just a simple tube. It starts with the Bowman's capsule. But we've got to add on our network of blood capillaries. So, as you know, the blood flows into the kidney through a renal artery. The renal artery will branch into lots of smaller branches called arterioles, and you'll get a little branch of that flowing into this area here, the Bowman's capsule. So it flows in there. And then basically you get this network, just a ball of capillaries, and then it leaves, blood leaves through another arteriole. The arteriole that's taking the blood into here is called the afferent arteriole, and the one that is taking it away from this ball of capillaries is called the efferent. Now, when I was in school, we had to learn Latin, and so these come from Latin words. So the afferent is taking it to the structure, and this structure, this ball of capillaries is called the glomerulus. So that ball of capillaries is called the glomerulus. The afferent arteriole is taking the blood into it, and the blood leaves it in the efferent arteriole. Now you'll notice that the afferent has a much bigger diameter than the efferent arteriole. Now if you think of people going into a hall and they go in through a massive entrance gate into this hall or this arena, people pile into the hall. If there's only one door and it's small and tiny to get out, that's going to build up into a big pile of pressure inside that building. So the same happens here. You get massive hydrostatic pressure in the blood vessels in the glomerulus, and that will lead to ultrafiltration, which we'll talk about in another video. That's something to look forward to. Now, so that's blood vessel, that's a branch of the renal artery, branches off into lots of arterioles. The afferent arteriole brings the blood into the glomerulus. Something happens to it there called ultrafiltration, and some of the liquid will pass into, it'll get filtered through here and into the Bowman's capsule. So the liquid that enters there is called filtrate because it's been filtered. So just like in chemistry, where if you've got a filter funnel and your filter paper, you pour your liquid in there, whatever drips through there is your filtrate. Hence, the liquid that manages to get into this cup-shaped structure called the Bowman's capsule is called the capsular filtrate. And so from now on, we talk about this filtrate. I'm not going to draw liquid in there. If you want to do a bit of colouring in, you could. So what I've done on my diagram, you'll see later, is I've actually shaded in the blood vessels to make them look different from the actual nephron. You could shade the nephron in yellow or something if you wanted, if you're bored, but I haven't got that bored yet. What about the other blood vessels? So you'll see in your diagram, you've got this big network of blood capillaries basically, and they're wrapped around this whole structure. And we've got them, branches of them branching all around here. And they basically wrap around the proximal convoluted tubule, the distal convoluted tubule, and we've got blood vessels as well that will go around the loop of Henle. Obviously I'm not going to draw it all, and then feed back into there. And then eventually this will become a branch, a flow into a branch of the renal vein. So that blood will flow into a branch of the renal vein. Lots of those will branch together and leave the kidney in the renal vein. Now, at this stage, we're not talking about all the different processes, but we've said that ultrafiltration happens here in the Bowman's capsule. That's due to the high pressure, and we get a lot of water, and we get a lot of things like glucose, a lot of minerals, magnesium, phosphates, salts, things like that, getting put into this filtrate. That will flow through the proximal convoluted tubule, and at that stage we get a lot of the water being reabsorbed back into the blood. And as you see, the blood's flowing around here and eventually will flow out of the kidney. So a lot of the water is reabsorbed here, but more importantly, all of the glucose that gets forced out here into the filtrate, all of the glucose, and if there's any amino acids and things like that, they will get absorbed back into the blood and leave the kidney in the renal vein. So we've got selective reabsorption happening here. It's not just everything, just certain things are selected for and they're absorbed here. The filtrate then flows down the descending limb of the loop of Henle and then up the ascending limb. Limb is just spelt L-I-M-B, just like your arm or your leg. It's like a limb going down and then up again. So you'll learn later that this loop of Henle, we've got fluid going down, fluid going up. That's called a counter current or a counterflow type system. So you sometimes in the motorway, if they've closed one carriageway, they have to divert one lane so that we've got the traffic flowing in opposite directions. It's the same here. It's not a counterflow, we call it a countercurrent mechanism. And that's set up so that the fluid, the interstitial fluid that is in between the cells in the medulla region of the kidney, it becomes more and more concentrated. And that has an effect later on when we talk about osmoregulation. So that's the loop of Henle. It sets up that increase in concentration as we move down through the medulla. Then we get to the distal convoluted tubule and the collecting duct. And this is where osmoregulation takes place. Osmoregulation is where the osmotic potential or the water potential of your blood is regulated. If you go out, you drink lots and lots of water or Coke or something like that, you'll end up running to the toilet a lot, produce a lot of urine, a large volume of dilute urine. That's because this is functioning properly and the hormones in your body are making the water leak out of here and get back in. I'll start that bit again. They're making the water stay in your urine and then be lost whenever you excrete. On the other hand, if it's a hot sunny day and you've been out playing golf or doing some kind of activity and you might even have been sweating a bit, your body wants to retain as much water as possible, and that happens here. The permeability of these cells will change and mean that you lose a lot of water into the fluid and it gets back into the blood. It stays in your body. It means that you produce less urine, a smaller volume of urine. But we'll look at the details of that later. So the main parts, Bowman's capsule with the glomerulus, that's where we've got a build-up of pressure and ultrafiltration takes place. Tiny molecules get pushed out of the blood and enter Bowman's capsule. Proximal convoluted tubule, that's where the useful substances like glucose and salts and so on are reabsorbed back into the blood along with a lot of water. That's where most of the water is actually reabsorbed. Then we've got the loop of Henle, and it works to create a very concentrated tissue in the medulla. Then we've got the distal convoluted tubule and the collecting duct, and they're responsible for osmoregulation, keeping the water level of your blood, the concentration, constant at the right level. So those are the main parts of the nephron. What I've done here, as I said, draw on the blood vessels, labeling them. I've labelled them in a different colour to show that they're the blood vessels rather than the parts of the actual tubule, the nephron. As we said, if we go back to this diagram here, this is where the nephron is. So we've got Bowman's capsule, the proximal convoluted tubule, the loop of Henle, distal convoluted tubule, flowing into collecting duct. The fluid or the filtrate there really becomes urine and it feeds into the pelvis region. Then the ureter is taken to the bladder and then leaves the body through the urethra. Okay, that's it for now.